Okay, class. Today what we're going to do is talk about microbes, and that includes the bacteria, which are the prokaryotic organisms, and we're also going to talk about viruses, prions, and viroids, which are not organisms, but as you can see now in the midst of the coronavirus outbreak, they cause a lot of problems. And then we're going to talk about protists, which are the single-celled organisms, and then we're going to talk about the fungi. Now, in order to begin, I just want to mention a couple of things to you right here. Firstly, is that bacteria are very important for the functioning of the planet, okay? And there is a tremendous quantity of bacteria on the planet, as we see here in the term biomass. Biomass means the total weight of these organisms. And we mentioned the total biomass on the planet. Of course, these are estimates. We put them in the gigaton range. 1 gigaton is equal to 1 billion tons, or each ton is equal to 2,240 pounds. Okay, so anyway, a metric ton contains 100 kilograms. Each kilogram is 1,000 grams. So it's roughly, each kilogram is 2.206 pounds. So anyway, as you can see, there are a lot of tonnage here. Plants, as you can see, is about 1.1 billion metric tons, because that includes all the trees, grasses, flowers, everything like that. The bacteria, which we're talking about now, they have a tremendous number, the second largest biomass on the planet. Again, these are all estimated at 500 gigatons. And then the protists, which are the single-celled organisms, such as algae and organisms such as malaria and things like this, they're called protists. That's only 10 gigatons. And then we have the fungi, such as mushrooms and yeast, which are 5 gigatons. And then if we would weigh all the animals together, including elephants, whales, snakes, worms, etc., roughly 2.5 gigatons. Now, bacteria come in 3 basic shapes. They come in round, rod, and spirochetes. And the rounds are called cocci, the rod shaped are called bacilli, and the spiral are called spirochetes. And here you see pictures of them. And these are all electron micrographs, scanning electron micrographs. This is computerized color. You cannot see real color under the electron microscopes. Now, I want to go into the functions of bacteria on the planet, and there are many functions. And let me show you what they are, because they play a major role in actually survival of life on the planet. And it's very important that you understand this. And we're going to put that as the benefits of bacteria. And benefits of bacteria are quite many. First of all, they fix nitrogen, which means they put it in a usable form that plants can use. They manufacture food, and they act as food also. We have them in cheeses and other things, such as sauerkraut, and food for other animals. They fight infections. They're used in drugs. They help treat diseases. They cause disease, by the way. I know, treating disease, we use that for experimentation. They cause disease. We use them in genetically modified organisms, such as GMO. They decompose organisms. A lot of them have enzymes that we use. Bioremediation means they break down hazardous waste and other substances, such as oil. Metabolites, such as vitamin B12. We need these for the bodies. They help in digestion. And of course, we use these in research. Now, before I begin into more detail, I want to show you a very good movie about bacteria, and this will be in the next section. As you're going to see right now, it has nice music, and I think you will learn a lot from it. minutes and they got it. So, just have to finish this. Okay, I want to continue now with bacteria. I want to show you this prismatic spring. This prismatic spring is located in Yellowstone Park, and it's very hot. Notice we have a lot of colors here, and all these represent bacteria. These bacteria are called extremophiles. Why? Because they live in extreme environments. Here, the environment is very hot. It's almost boiling. And over here, you see a boardwalk, and you'll see all these people walking there who are interested in looking at the prismatic spring in Yellowstone. And I'm going to continue with this. I want to show you that we have other types of extremophilic bacteria. These live 2 miles down in Greenland. Remember, every 1,600 meters is 1 mile. Every 1,000 meters is a kilometer. So this is over 3,000 kilometers deep. So if this were 3,200, I'm sorry, this is 3,000 meters deep. And if this were in miles, if this were 3,200 meters, it would be almost exactly 2 miles down. So here you see these bacillus types of bacteria. And I want to point out that when we study bacteria, we also mentioned something called blue-green algae. These have a new name called cyanobacteria, because they're blue-green and they contain cyanophycin, which is a blue-green pigment. Many of these are toxic, and right now we're writing another book about algae, and we mentioned these blue-green algae in the toxic section of the book, well, in the section about toxins, actually. And one of the most famous species is called microcystes, and this is one that was contaminating the water in Ohio. That's right, it was contaminating drinking water in Ohio a number of years ago. And over here, let me just show you some more pictures of bacteria. These, of course, are bacilli taken with a scanning electron microscope. Here we see a chart which lists the various organelles, but I'm not going to go into this one. I'd rather go into this one because this is short and sweet and to the point. Here we see a transmission electron micrograph. Here we see the bacterium, that one bacteria. Over here we see a plasmid, which is a circular piece of DNA that I'm going to talk about in a few minutes. And then I'm going to show you this nucleoid, which instead of a nucleus, since they don't have a membrane surrounding the single chromosome, the nuclear region is called a nucleoid. Here we have an inclusion. And over here we have a plasma membrane, which of course is found in all cells. Then we see something called a cell wall, which is found in most bacteria. And then we have a capsule, with a mucus-like material. And this prevents the immune system of an organism that attacks it from gobbling up, because it'll just slide under it. And here we have a flagellum. And again, this is a conglomerate of various types of bacteria. I want to point out, though, that the cell wall of the bacteria is not exactly similar to the cell wall of plants. So anyway, I just wanted to point this out. Okay, now, if we look here, we'll see a microscope. And in order to see bacteria, you put it on the 100-power lens, and it has to be a 100-power oil immersion lens. And we put oil on the slide, and what the oil does is refocus the light so it goes straight instead of crooked. Therefore, you can see the organism better. And this next picture shows you the bacteria, which has a plasmid. And what happens is the plasmid is being transferred into this bacterium here, and then that's going to divide as they reproduce, and I'll go into that in a few minutes. And then we see what happens here is the genetic material will be the same in both these bacteria. Here's another bacterium. Now, what I want to do is play a little movie on plasmids for you, which I think you will find interesting.
Bacteria have been helpful to genetic engineers, not only in providing them with restriction enzymes, but also with what are called plasmids. In addition to their single chromosome, many bacteria also contain tiny rings of DNA called plasmids. Plasmids are usually about 1,000 to 100,000 nucleotides long and act as independent, self-replicating molecular operators within bacteria. Plasmids are one of the ways that DNA recombination goes on within nature. While a bacteria's chromosome contains all the necessary genes to code for the bacteria's existence, plasmids provide an effective way by which traits not contained in the chromosome can be passed from bacteria to bacteria. For example, some plasmids contain genes that code for enzymes that digest certain antibiotics, such as penicillin or ampicillin. This is obviously an advantage to the bacteria. When a bacteria containing these plasmids dies, it breaks open and liberates these plasmids to the outside environment, and they are often taken up by other bacteria that then acquire the traits coded for by the plasmids. Genetic engineers can place liberated plasmids in a solution with restriction enzymes and cut them apart. The engineers then place the gene segment they wish to insert into the solution with the plasmids, and then add repair enzymes that join the pieces of DNA together at their sticky ends. These new plasmids, with their newly inserted genes, are then placed with bacteria into a different solution that enables the plasmids to readily penetrate through the cell wall and membrane of bacteria. The bacteria that are exposed to the plasmids are taken out and grown in a culture for a short time. If the plasmids into which the experimental gene, such as the gene for the production of insulin or growth hormone, is inserted, also have genes that provide resistance to ampicillin and amoxicillin, a unique technique can be used. Genetic engineers know that not all the bacteria exposed to the plasmids will absorb them. By exposing the cultured bacteria to antibiotics such as ampicillin and amoxicillin, they kill off the bacteria that didn't absorb the new plasmids, while those bacteria that did absorb the new plasmids containing the genes for antibiotic resistance and for, say, insulin or growth hormone continue to grow and thrive in the culture. Scientists continue to grow these bacteria until the insulin or growth hormone produced by them is sufficient to be extracted and purified for use by human patients. But how are genes inserted into the chromosomes and DNA of more complex organisms such as plants or animals? There are two major methods of placing DNA into the nuclei of eukaryotic cells: vectorless and vector methods. Vectorless methods of insertion of DNA don't require the use of a third intermediate organism to deliver the DNA from one species to another. Vectors, in the biological sense of the word, are organisms that carry the DNA of one species and insert it into another.
Okay, now we're back. And so deadly bacteria are known to resist drugs, and that is because they had plasmids inserted into them. And like I said, this is a good form of research now for many things. Here we see a mouth. I don't think anybody wants to kiss people that have swollen tonsils here or the pseudomembrane caused by bacteria. Let's go into this. I just want to mention briefly something called the Gram stain, because this has to do with the cell wall of bacteria. There are two types of cell wall of the general types of bacteria that we mainly deal with. There's another type, which I'll get into in a moment, of which tuberculosis has. The Gram stain is a stain which stains the cell wall of a bacteria, and they use certain dyes. And if the end result is purple, that is called Gram positive. If the end result is pink, it's called Gram negative. And the doctor can determine what kind of drugs will work on the bacterium depending on the cell wall. Now, if we look over here, here we see the cell wall of Gram positive and Gram negative bacteria. A Gram positive bacteria has more of a substance called peptidoglycan, whereas a Gram negative bacterium has something called the lipid layer, which is fats. And these fats dissolve, and so at the end we put a counter stain, which gives it a pink color. Okay, so a purple contains peptidoglycan because it holds the dye. Lipid layer here, which is the fat layer, loses the dye, and we have to put a counter stain or a counter dye, and that gives you a pink color. Now, let me show you these two pictures here. Here we have a electron micrograph of a Gram-positive bacteria, and the Gram-positive bacteria has something called peptidoglycan, which is a substance, and it's quite thick in a Gram-positive bacteria, as we see here. And then this next picture is a Gram-negative bacteria. Here we see less peptidoglycan, but more lipoproteins here, which are fats. And here we see it again, a transmission electron micrograph of the bacterial cell wall. Over here we have something called the acid-fast stain, where organisms like those that cause tuberculosis and leprosy are members. The organism which causes tuberculosis, it's a bacillus called Mycobacterium tuberculosis, and the one that causes leprosy is Mycobacterium leprae, and they use something called an acid-fast stain, which is quite different than the other two. Here we see one of the largest bacteria in the world called Thiomargarita namibhiensis. I think you can almost see it with the naked eye without a microscope. Here we see electron micrograph of, this is a scanning electron micrograph of cocci. Here we see a light micrograph of cocci, and you can see the difference between this, how clear it is, and here it's slightly blurred, but a trained microbiologist or bacteriologist can know the difference. These are bacilli. They live in chains. They're connecting, something like long sausages. Here we have color-coded bacilli taken with the scanning electron microscope. And no, that is not macaroni and cheese, although it looks like it. Here we see another bacillus. Here we see on the right the cross-section of a transmission electron micrograph of a bacteria, on the left of a bacilli. On the left we see a scanning electron micrograph. Here we see a light micrograph of bacilli. Scanning electron micrograph of sporillum, light micrograph of sporillum. Again, another sporillum, probably a light microscope. Look at this dancing sporillum taken with the scanning electron microscope. And here we see, of course, the upper pictures are those taken with the light microscope. The bottom with the scanning electron microscope. And over here we see that's a sporillum with the flagellum. Here we see moving. You might call this a moving experience of the sporulum, actual photographs. Here we see a couple photographs. I like the bottom one. It says the most important thing in life is not letting the little things get to you. And in the upper one, you see a small microbiologist at a bar talking to this lady who happens to be smoking, which of course she shouldn't do. Over here, we see on this top picture, we see something called mycoplasms. Mycoplasms are bacteria without a cell wall, and that actually makes them more difficult to treat if they cause disease. Here we have some more mycobacteria. These are also called L forms because they were discovered at the Lister Institute in England many years ago. Again, by not having a cell wall, it makes it easier to penetrate a little hole where they can do damage in an organism. And here we see some colored light micrographs. The top is a sporulum, the bottom is a flagellated bacilli. Of course, the top is a flagellated sporulum. Here we see, I believe, is anthrax, an infection on the skin. And on the right, we see actual micrographs of anthrax. Again, these are bacilli. Now, how do bacteria multiply? Well, I'm going to talk to you about three ways. One is done by spores. Another is binary fission, which is probably the most common way. And another one was found by an Israeli scientist of a large bacterium called E. policium, in which it reproduces similarly to humans in that through a slit, little baby bacteria come out. Anyway, let me start with this called spores. How does sporulation occur? Well, what happens is first we have the nuclear material in the bacterium, which of course is one chromosome, and that's going to divide, as we mentioned previously when we discussed DNA and the chromosome replication. And then what's going to happen, a plasma membrane is going to surround the DNA and the cytoplasm. And then a septum, which is another structure, is going to surround that, as we see here. And then what happens, peptidoglycan, as I mentioned before, happen to be in the cell wall a lot. You have a lot of it in the gram-positive bacteria, is going to form here, and then the bacteria is going to be released. The spore is going to be released from the bacterium, and then that will float in the water until environmental conditions are right for it to germinate. And germinate means to grow, not migrate to Germany. And here we see a transmission electron micrograph of the spore, since it's on the inside of the bacteria, called an endospore. And here we see transmission electron micrographs of the spore, as we see here. And I'm going to show you spore formation. And I'm going to have to see if I can get this. This is going to cause me a problem because we need a some sort of plug-in. So I have to go to a different, let me get a different, let me see if we can go to Google on this one. Oh, that was Google. Let me go to, let me try Google. I have to put in a plug-in. I don't think we have to find that now. Let's try it. Let's keep our fingers crossed. If it doesn't work, I can't show you. And let's see what happens. Nope, that doesn't work either. So let me try one more thing. Let me go back to Brave here. See what happens. Nah, that's not going to work. Try one more time. And that doesn't work. All right, I'll have to go back to that later. I'm not going to worry about this now. Let's continue. I will try to get that movie later. Okay, now, if you look at these cans here, what you see there are the cans are swollen. Why are they swollen? Because they contain something called anaerobic bacteria, which means they grow without oxygen. And the way you can something is you put the bacteria, you put the food in the can, and then you cook it in the can. And what happens is if they have spores, the spores are going to germinate, which means they're going to produce a gas and grow, because many of these are anaerobic, which means they can live without oxygen. And and that causes a gas of carbon monoxide there. And I like this picture here. It says, don't worry, that happened to me too, but botulism got rid of the wrinkles. And one of the substances here is caused by Clostridium botulinum that I will talk about in a few minutes. And here we see some other cans, okay? And what I want to do is show you how these bacteria multiply, and I will get to that in a moment. Let me get here the bacteria multiplying. I'll have to go back here for a minute. Called binary fission. Let me bring this up, and we're going to get some binary fission. I thought I had that there. Binary fission. It should have worked before. Let's go to the video here, and this is the way they should work. Let's see here. I got to find binary fission video. There we go. Let's see. I'll have to go to a different one here. Let's go to YouTube. I think YouTube might have it. I did have it here, but somehow it got discombobulated. All right, let's go to YouTube, and we should have it here. Just bear with me. These things take time. And bacteria, let's see what happens. There we go. Let's see if this works. That's what I want. Let's try this. This one's going to work, and go. Yeah, there it goes. If you look closely, all this originated from one bacterium. And what happens is they keep multiplying. And I'll show you how this works in a moment. All that from one bacteria. Look at that. Isn't that great? Now, what I'm going to do now is we're going to get out of this one. Let me get out of it. And I'm going to go back to the PowerPoint here. And I'm going to show you this. Let's get this working. There we go. Now, essentially, how does this work? Well, it works like this. You have a bacterium. Again, the nuclear material is going to double, and then the cell splits in two, and the end result is two new cells. And let's see here. In one day, you can get 5,000 billion bacteria, which means you can have 1, 2, 3, 4, 5, 6, 7, 5 times 10 to the 21st power, which means you have 21 zeros. And as you can see, that's quite a large number. So that's what happens when bacteria multiply. But of course, they can't because they're limited by nutrients. They can't keep going on. Okay? And here we see another picture of how the bacteria divides. And that's the way it is. So that's how these things divide. And by the way, if we look at this picture, this is one out of every 10,000 bacteria exchanging genetic material. These scientists took this picture where evidently one of these organisms is exchanging genetic material. I have no idea which is exchanging, who's exchanging to whom. Could be this is going this way, there's a pilus, or this is going this way. I have no idea. But anyway, you get the idea. Now, as for the Epuliscium, this is the organism in which a baby bacteria come out. Now, how does that work? Well, notice this is a large bacterium, and here we see an E. coli. I believe this was discovered by an Israeli scientist, and here we see some Epuliscium. And in lab, remember we looked at paramecium under the microscope. The paramecium were quite large, and if you look at these right here, you'll see how big this bacterium is compared to the paramecium, which are quite large as a rule. And what happens is there's a little slit inside the bacterium and the baby comes out, similar to humans. And if you look at this right here, here we see a little baby coming out. So it's quite interesting, but I'm not going to dwell on that. Now, what I want to do is, oh, here we see another picture of the Epulopiscium where a little baby comes out. Okay, here we see a whole conglomerate of various types. And now what I want to do is talk to you about anaerobic species. These are the ones that cause the botulism, as I mentioned before. These are the four major ones, and anaerobic means they live without oxygen, in contrast to the word aerobic. Okay, so if we look closely, we see some diseases of these, and notice the first one I put is Clostridium botulinum, and it's a very bad toxin, which means it's a poison, but doctors use it to get wrinkles in foreheads. Women use it. They give them shots to get rid of wrinkles. We also have Clostridium tetani, which causes tetanus, obviously also called lockjaw. That's a very bad one. You can't move your jaw. Clostridium perfringens causes gas gangrene, which is not good. And then C. difficile. This is a very common one found in hospitals where workers, such as orderlies and other people, nurses, might become infected because they're dealing with patients. And I once had a student who was sick for this for about 3 weeks. So fortunately he survived, but it's not a good one to have. And again, these cause these very bad diseases. And if we look at this picture, we have various types. Let me just show you here. We have something called facultative anaerobes, and what that means is a facultative anaerobe is an aerobic bacteria that can live both with oxygen and without oxygen. And if we look at this tube, we will see you have some growing here near air and some growing without air. But when we have obligate aerobes, the obligate they must have air. And if you look at this test tube here, of course, these are drawings, but nevertheless, you get the point where there's nothing down here because they need oxygen to survive. But then you have obligate anaerobes, and they live at the bottom because there's very little oxygen there, possibly none, where here there's a lot of oxygen. And then you have aerotolerant, which means they can live with air and maybe without air. And then microaerophils need a very little bit of oxygen. And this is a petri dish where we grow most bacteria. And here we see another type of bacteria. This is an intracellular parasitic one. They do not live outside the cell. They live inside the cell, and these are called rickettsias. And rickettsias are gram-negative intracellular bacteria, and a very famous disease caused by this is Rocky Mountain spotted fever, which is you get from the bite of a tick. Here you see some ticks. And what happens when you get bitten by a tick? You can get a few diseases. Lyme's disease, which is caused by a bacterium, and also Rocky Mountain spotted fever, which is caused by, again, the rickettsia, and it's bitten by a tick. And I should point out that when you get bitten by a tick like these, you get ticked off. Just thought I'd mention that. Okay, one more thing before we go on to viruses. I just want to show you the way these organisms, there are certain bacteria which live at the bottom of the ocean, and they act as food by giving off energy to other organisms, such as the deep sea vents, where you find a lot of giant crabs and worms and things like that there. And notice the formula for photosynthesis is CO2 plus water, and that gives you a sugar. However, underneath the ocean, instead of using water as a hydrogen acceptor, they use hydrogen sulfide. Of course, they're in the water here, and hence you get the same sugar, but you get sulfur compounds. And it is this which supplies the energy to the organisms which live in the deep sea vents, such as large worms called tube worms. And we'll go more into that when we discuss nutrition types when we get to the protists. And here we see these worms. That these are the tube worms. They live without stomachs because they eat they don't really eat since they have no stomachs. They get the energy derived from the bacteria. And I'm going to stop here for now, and then we'll talk more about the viruses next. Okay, class, I noticed that in the original first lecture that I gave, I couldn't get the spore movie or the binary fission moving working. So what I want to do now is finish up with this, and then I will put a new section about viruses, and then after I do the section on viruses, I'll talk about protists, and then after I put the protist section, I'll talk about the fungus. Okay, so what I'm going to do now, I finally got this working on a different browser. I'm going to play this for you right now. This lasts a couple minutes about spore formation. So here we go.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolenic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell and the mature spore is released.
Okay, now what I want to do is show you binary fission, okay? And I'm going to show you how this works. And this is pretty cool. I think you will enjoy it. And this is important. I managed to get this one working too. So let's see if we can pull this off.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
That's right. 5,000 billion is 5 times 10 to the 21 power. Now, I want to point out that this is binary fission, not to be confused with fishing. This is fishing, just so you clarify that. Okay, I just wanted to verify there's a big difference.